Hi everyone, and welcome to this journey into the Health and Life Sciences building, hosting the School of Biological Sciences at the University of Reading, a beautiful and diverse environment for learning and research. My name is Susanna, and I'm a member of the Alzheimer's Research UK Thames Valley Network. I recently moved to Reading from the University of Padova in Italy to join the laboratory of Dr. Eva Kevei and work on a very exciting project that has to do with neurodegeneration and understanding what predisposes us to develop certain age-related disorders, such as dementia and Parkinson's disease, in collaboration with Dr. Patrick Lewis at the Royal Veterinary College in London. Reading is a beautiful environment to do this, because we have a very active and collaborative neuroscience community across multiple schools, and I have the possibility to interact with a lot of excellent scientists with different expertise. So the plan for today is to tell you a little bit more about our research, what we do in our laboratory, and how I try to answer some key questions that could help us further understand Parkinson's disease and dementia. Key questions that I keep in mind every day while undertaking my experiments in the lab. In our laboratory team, we all have different interests and expertise, but one aspect that we have in common is that we are all very intrigued by understanding how the brain works, what happens during the process of aging, and what goes wrong during brain diseases. In doing so, we study a very simple but very powerful microscopic transparent world called Cenorhabditis elegans, or C. elegans for short, which can act as a simple simulation to better understand human diseases. C. elegans is one piece of the puzzle in helping scientists unlock the current mysteries in neurodegenerative conditions. For example, we have known for many years that when a person is affected by a neurodegenerative disorder, some precious cells in the brain, the neurons, die. But we still don't understand what the primary cause of their death is. For these reasons, thousands of scientists all around the world are researching dementia and neurodegenerative diseases in hundreds of different ways. Some teams study different simulations, such as brain cells in a dish or fruit flies, for example. Findings obtained from each of these investigations are compared with each other, and if similar, we as scientists can better pinpoint what is happening to cause dementia, Parkinson's, and other neurodegenerative diseases, creating a fuller picture from the individual puzzle pieces. Through having a better understanding of conditions linked to dementia, new and better treatments to help manage, improve, or ideally prevent these diseases could be developed in the future. A lot of research in our laboratory is focused on further understanding Parkinson's disease, a common and often age-related neurodegenerative condition. Like Alzheimer's disease, it is a progressive condition, which usually worsens over time. A person with Parkinson's develops a number of symptoms related to their movement, but also often have much broader no-movement symptoms. This can really impact a person's life and well-being, and they are often different for every person with Parkinson's. Some people with Parkinson's develop memory and thinking problems later in life, with many developing dementia later in the progression of the condition. The symptoms of Parkinson's are caused by the loss of neurons which produce and use the chemical dopamine to communicate. Currently, many drugs to help Parkinson's movement symptoms focus on replacing the dopamine lost. These are often very effective, but can become less effective as the condition progresses, and we currently have no drugs to stop or slow this from happening. Through scientists developing a better understanding of what is going wrong to cause the loss of dopamine neurons in Parkinson's, new treatments could eventually be designed. In very rare instances, Parkinson's can run in families, and the responsible genes cause the disease strongly. A lot of the progresses that we have made in understanding what happens in the cells of people who develop Parkinson's come from the extensive study of these particular genes by hundreds of laboratories in the world. In contrast, most cases of Parkinson's have no known cause, but we are beginning to understand that there may be many common genes that slightly increase the risk of developing the disease. Through understanding what happens in genetic Parkinson's, we can better picture what happens in the cells to cause the condition. This is the major aim of my research project. I am particularly interested in defining what are the changes in the DNA that contribute most to Parkinson's disease predisposition. Recent genetic studies show that the majority of people with Parkinson's likely have alterations in multiple genes that together slightly increase the risk of developing the condition. These studies suggest that one possible common cause for Parkinson's risk is a general dysfunction in the cellular recycling system, whose efficiency is essential to clear the materials that are no longer required within the cell, as well as any toxic species. C. elegans are particularly powerful for the study of neurodegenerative disorders, such as dementia, because they grow very fast and we can see how the neurons develop and are affected by aging. C. elegans grow from an egg to an adult in 3 days, and signs of aging can be seen and studied just a few days after this. Humans have around 86 billion neurons in the brain and are incredibly complex to study, while C. elegans have just 302 neurons in their whole body. Hence, studying the simple neuronal system of worms will provide us an easy-to-use platform to understand how neurons affect a whole animal and what happens to them in dementia or other neurodegenerative conditions. In the last few decades, scientists studying C. elegans have understood exactly what each of these neurons does and what chemicals it produces, like dopamine, which is relevant in Parkinson's research. I know that it is incredible, but little does not mean simple, and we can learn a lot from studying these tiny animals. Also, C. elegans have many genes in common with humans. For 60 to 80% of human genes, there is an equivalent similar gene in C. elegans for us to investigate in the lab. As we spoke about earlier, we know there are some genes relevant to Parkinson's and other neurodegenerative conditions, and many of these can be studied in C. elegans. We can even introduce the subtle changes in DNA seen in a small number of people with inherited forms of Parkinson's into the DNA of the C. elegans to better understand the effect of these changes. This is what my colleague Rachel Chandler, Parkinson's UK PhD researcher and fellow member of the Alzheimer's Research UK Thames Valley Network, has been working on for the last 3 years, similar in many ways to my project. Come and meet her.
Hello, thank you for coming up to look around the lab. I'm going to show you and talk to you about just one of the ways we can study C. elegans and how they can be relevant in furthering our understanding of Parkinson's. As Susanna mentioned, C. elegans have 302 neurons. We understand what each of these neurons does and what chemicals they produce. C. elegans have 8 dopamine-producing neurons. 6 of these are found in their head, and they control a behaviour that helps the C. elegans worm find a food source. Our C. elegans in the lab live on Petri dishes, which are coated with a bacteria which isn't harmful to the worm, instead they eat it. When C. elegans are added to a Petri dish that doesn't have any bacteria, their only food source, they crawl around fairly quickly to increase their chances of finding food. We add worms to a Petri dish with bacteria, just coating a ring around the outside. If the worm's dopamine neurons are working correctly, when they reach this ring of bacteria, they will slow down immediately, stop, and have a feast of tasty bacteria. If the dopamine neurons aren't working as they should, the worms have a delay in or do not slow down. We film the worms doing this, track their speeds with a computer program, and calculate how much they have slowed down compared to their usual speed. I'm going to show you a quick example of this and how we've used it in an experiment in the lab. Here, we have tested a worm population which has a rare Parkinson's gene change engineered into its DNA. As you can see in the dark blue column, it only slows down by around 65%, compared to worms without this gene that slow down by around 95%. This is amazing, as it shows that a gene that causes Parkinson's in humans also has an impact on the worm's dopamine system, despite C. elegans being so simple and so different to humans. If we feed the worms dopamine before running this test, shown in bright blue, this improves the population carrying the rare Parkinson's gene to slow down much more and behave very similarly to worms without this genetic Parkinson's change. This is also so interesting that we see this in the worm too, as current treatments for Parkinson's, which help manage movement symptoms, often involve drugs that replace the dopamine lost in the brain, somewhat similar to what we have done here. In the fact that we see something similar in the worm helps us further pinpoint that they are a promising new simulation to better understand the condition. We can further develop our understanding of what's going wrong in the neurons through many different forms of microscope work, which Susanna will tell you more about now.
As Rachel mentioned, we can utilize some very potent microscopes to have a closer look at the dopamine-producing neurons and verify what is the reason why we observe those alterations in the behavior of the worms when searching for food. Using very advanced genome engineering techniques, we can introduce a fluorescent gene from a jellyfish into the worms to light up their 8 dopamine neurons for us to see them using potent lasers and microscopic lenses. In this way, we can check whether they are healthy or they show any signs of neurodegeneration. Here, you can see the comparison between the worm without any mutation, showing an intact and healthy network of dopamine neurons, versus a worm carrying the Parkinson's gene change described before, having very unhealthy and degenerating neurons. This is very intriguing because by combining the test described by Rachel and the work that we can do at the microscope, we can understand what the cause of alterations in how an animal behaves is. But every cell is like a little town, and there is a lot going on inside. Exploiting the high resolution of our microscopes and some very powerful dyes that can be very easily administered as food to the worms, we can enter the cells and take a look at the subcellular compartments, the organelles. In particular, I am interested in the compartment that takes care of recycling the waste material within a cell and clearing up the toxic species, the lysosome. As we mentioned before, the happiness of the lysosome is fundamental to maintain the whole organism healthy. And a lot of studies have shown that maintaining lysosomes functional is associated with longevity. On the other side, when the lysosomes do not work properly and their ability to degrade materials is lost, the risk of developing neurodegenerative disorders increases. In the lab, we have several ways to measure if lysosomes are functioning correctly. One example is using dyes that change their color depending on how well the lysosome is working. Here, a yellow signal means that the lysosome is working properly, while a red signal means that something is going wrong. For us, having a precise indication of what is not functioning correctly inside a cell affected by a neurodegenerative condition is essential, because it represents the only way to try alternative strategies for correcting the alterations. We have discussed a little bit about how single Parkinson's changes in the DNA can have an impact at many levels, and how studying the worm as a simulation can help in further characterizing this. However, almost nothing is known about how different genes interact with each other and lead to Parkinson's. Using C. elegans, I have the possibility to reproduce the situation that happens in people with Parkinson's and look at whether combining mutations in other genes makes the problems worse, stay the same, or get better. Once I will find the most dangerous combinations, I will give the worms some drugs or switch on and off genes in order to try and correct what is going wrong. Moreover, I'm planning to utilize a similar platform to study other neurodegenerative diseases, including various subtypes of dementia. These findings will guide future studies on human brain cells and may aid the development of new drug treatments. Thank you very much for staying with us today. I hope to meet you soon at Reading.